let's continue with chapter 22 and look at what some of those receptors on the T cells do. So remember that T cells have TCR and CD receptors. First, we'll look at the cytotoxic T. So cytotoxic T cells have TCR and they have CD8. Those are the two proteins that we talked about before on the surface of a cytotoxic T. Over here, we will look at a body cell. Okay, so this is the inside of a cell. And here you can see this is the Golgi, right? And here's vesicles. Hopefully some of this is coming back to you from chapter 4. This cell is healthy. And cells of the body have to carry ID. Your immune system has to be able to identify cells and know who they are. So these things that you see here are the cell's ID. It has two parts. The small orange part that you see is called MHC. This stands for Major Histocompatibility Complex. Then the little purple dot on top of it is antigen. So just like pathogens have antigen, just like your red blood cells have antigen, all of your cells have antigen on them. That's how you identify your cells. In a healthy cell, it is self-antigen. So you have your own unique antigen. It's encoded in your DNA. It's unique to you. And that's on the surface of your cells. So a cytotoxic T will interact with a body cell. The CD8 will bind to the MHC1. So body cells have MHC type 1. The TCR will then inspect the antigen. Let's look at the next cell. This one is an infected cell. Okay, here you can see, here's the pathogen. The pathogen has entered the cell and the cell is sick. It's infected. Notice that the MHCs are different. If you look close, see the pathogen has little kind of teal colored dots on it that are its antigen. Look over here at the MHC. There's that pathogen antigen on there. So in an infected body cell, you have your MHC type 1, and then you display pathogen antigen on it. So let's look at what happens when a T cell encounters a body cell. Here is our T cell, so this cytotoxic T, and this is a body cell. So a cytotoxic T cell will encounter a body cell. The first thing that happens is the CD8 of the cytotoxic T will bind the MHC1 of the body cell. The second thing that happens is the TCR inspects the antigen. Okay, so by inspecting the antigen, it's looking at the cell's ID. If it is self-antigen, or it is pathogen antigen, but it does not match this particular T cell, then the T cell will disengage and leave it alone. Okay, but if it is pathogen antigen on that MHC, and it does fit, and they fit together like puzzle pieces, if the pathogen antigen on the MHC fits the TCR on this cytotoxic T, then you activate that cytotoxic T. So there's some more detail about how the two receptors on cytotoxic T work. So cytotoxic T interacts with body cells. The CD8 binds to MHC1 that's on all your body cells, and then the TCR inspects the antigen. Okay, now we'll look at helper T cells. We'll start off with this top cell here is an antigen presenting cell. We can call it APC for short. There are 3 cells in your body that are eligible to be antigen-presenting cells. These are B cells, dendritic cells. You might recognize this from Chapter 6. We talked about epidermal dendritic cells and macrophages. What these cells have in common is they are all phagocytic. This means that they engulf pathogens. It's like Pac-Man, going around engulfing the little dots. These cells go around and engulf pathogens. So here is the pathogen. And here you can see it's entering the cell, but it's not infecting it. The cell is eating the pathogen. So this is not an infection, it's the cell eating the pathogen. Then here you see the pathogen antigen gets displayed on MHC. So notice the pathogen has those little teal antigens. There's the teal antigen being displayed now. This is MHC type 2. And this is your pathogen antigen. So after a B cell, dendritic cell, or macrophage engulfs a pathogen, it will then display the pathogen antigen on its MHC2. So let's look at what's happening here. Here is our helper T. And here is our antigen presenting cell. The antigen presenting cell has pathogen antigen on MHC2. So a helper T encounters an antigen presenting cell. First thing that happens is the CD4 on the helper T will bind the MHC2 on the antigen presenting cell. Then the TCR inspects the antigen. Okay, it's not going to be self-antigen. Antigen-presenting cells present pathogen antigen. So we don't have to worry about if it's self-antigen. We just need to see if this pathogen antigen matches that particular helper T. Remember, you have lots and lots of different TCRs that you inherit from your parents. We have to find the one that matches, like having a bucket of keys and finding the one key that'll unlock the door. So if the TCR does not match the antigen, then they just disengage and they go along. If the TCR does find the antigen, or does fit, then you activate this helper T. Okay, so helper T cells only interact with antigen-presenting cells. They don't interact with body cells. They don't interact with the pathogen directly. Only these three types of cells that can have MHC2 will interact with a helper T. What we have just gone through now is basically the antigen challenge. So your third line works in 3 steps. It has antigen challenge, clonal selection, effector response. Okay, so kind of go through and we'll put all this together. We'll start off with, just to see what color it had, the light purple. Here is an antigen presenting cell. So our antigen presenting cell engulfs a pathogen. Then it displays pathogen antigen on MHC2. Then we have our helper T. So the antigen presenting cell will go up to a helper T. The helper T, CD4, binds the antigen presenting cells, MHC2. Then the TCR inspects the antigen. This is a trial and error process. You just have to go up to each helper T and do this process of the CD4 binds the MHC2, then the TCR inspects the antigen until you find your match. This is why it's a delay in the third line, because it might take a few days of trial and error before you find that match. Once you find the match, you activate this helper T. Okay. This part is antigen challenge. So what we have described here, this is antigen challenge. Now that you have your matching helper T, we need a whole army of these helper Ts to fight the infection. So now we're ready for clonal selection. So the helper T, notice, has chemicals that come out. and these chemicals loop back around to the helper T. This is interleukin-2. So an activated helper T secretes interleukin-2. And it secretes this chemical back onto itself. This then stimulates the T cell to clone so that you get clones of the helper T. This is the clonal selection. So this is step 2, clonal selection. Okay, in the meantime, body cells have been getting infected. So here is an infected body cell. And remember, cytotoxic T interacts with body cells. So now we have a cytotoxic T encounters an infected body cell that is displaying pathogen antigen. And this would be displayed on the MHC1. Okay, so again, it's that trial and error. You have lots and lots of different cytotoxic T cells. You have to find the one whose TCR matches this antigen. So first you will have the CD8 bind the MHC1, then the TCR inspects the antigen. And you keep repeating this process until you find your match. So there is your antigen challenge. Okay, then once you find your match, you have to sit and wait for a helper T. So here are your helper T clones. Notice the helper T clones are secreting chemical. They're secreting interleukin-2 onto your activated cytotoxic T. So once you find the match, then you have to wait for a helper T clone to come along and secrete interleukin-2. And then this stimulates your cytotoxic T to clone. So now here are your clones of cytotoxic T. So this is your clonal selection. Okay, so you're building up that army. Okay, let's look at the B cells now. We haven't talked about those yet. Here is a B cell. And remember, it doesn't have any CD. We just have the BCR that interacts directly with the pathogen. Remember also that B cells can be phagocytic. They can be antigen-presenting cells. So here we have a B cell encounters the pathogen. Okay, it will engulf it. It will then display the pathogen antigen. on MHC2. So it is now an antigen-presenting cell. Antigen-presenting cells go to helper T. So here is our B cell antigen-presenting cell. It goes to helper T. And remember, there's specificity here. Only the helper T, whose TCR matches the antigen that this B cell is displaying, will work. You can't just go to any helper T. So then the B cell will encounter a helper T. The CD4 binds MHC2. Then the TCR inspects the antigen. Might take a few days of trial and error until you get your match, but then you find your match. So this is antigen challenge. Once you find your match, not math, match, then the helper T secretes interleukin-4. And this stimulates the B cell to clone. So now, because of that interleukin-4 from the helper T, the B cell clones. Here are your clones of the B cell. Notice you have two different types of clones. These clones are memory cells. You're going to keep these, you're going to set these back and save them so that if you ever have this same pathogen again, you can already have an army ready to go. These are plasma cells. And you notice all of this here is antibodies. Plasma cells make antibodies. So this part here is your clonal selection. Okay, I know this particular set of notes is kind of a lot to take in. You might want to rewatch this video, and we'll go over this in class too to help make sure this is all making sense. But a summary, we put a summary so far of what we did here. You have cytotoxic T interacts with body cells. CD8 binds MHC1 and TCR inspects antigen. Then we have helper T interacts with antigen presenting cells. CD4 binds MHC2, then TCR inspects antigen. B cells will engulf the pathogen. If the BCR matches the pathogen antigen. So a B cell can't just engulf any pathogen. It has to be the B cell whose BCR matches the pathogen antigen that engulfs it. Then your helper T secretes interleukin-2, both onto itself and on to the cytotoxic Ts, and that's how you get your clones. And it secretes interleukin-4 onto the B cells. And that's how you get your clones. And then remember that some of these clones become plasma cells. Alright, so there is the antigen challenge and clonal selection of the third line. In the next video, we'll go over the effector response. That's what these cells actually do to go out and fight the infection. So this part was antigen challenge. You have to find the match. You have to find the TCR or BCR that matches the pathogen antigen. And then clonal selection, once you find those matching cells, you clone them to make an entire army of them.